Welcome to the third and final lecture on Chapter 10, and we're finishing 10.3 and moving into 10.4. So just to conclude section 10.3, some of the medical applications of DNA technology include gene therapy, which is a collection of different techniques that are used to identify defective genes and replace them with healthy genes, or to increase or decrease the expression of the defective genes. And gene therapy can be done in vivo, meaning in cells that are within the body, or ex vivo, which is where the cells are removed from the body and then modified or altered. And there are several proposed treatments that involve silencing genes with regulatory RNA, but they're not approved yet. This table 10.4 from your ebook includes some of the applications that are derived from genetically modified or transgenic animals. There are a whole bunch of them treating human blood clotting disorders using genes from pigs, replacing protein to prevent emphysema, using genes from sheep, stimulating the production of T cells to fight cancer using rabbit, and then iron binding protein inhibiting bacterial or viral infections from cows, using cows and human albumin in surgery to keep blood volume up, and then also dissolving blood clots or reducing heart damage using goat or tissue plasminogen activating factor. There's another one from goats that is antithrombin, and there's this drug Atrin, which was the first drug that was created from a genetically engineered animal that was approved by the FDA, and that's used to prevent blood clots. And there's a whole lot more treatments that have been derived from transgenic animals, including, malaria resistance from mosquitoes, using goats to secrete silk protein in their milk, pigs, and their pork meat. There are enzymes in the saliva of enviro pigs that are used to process excess phosphate in the feed, reducing pollution. Mice, cats, pigs, monkeys, and tropical fish are research animals that have been engineered with green fluorescent protein from jellyfish and causes their cells to glow green in UV light. These animals are used to trace development in embryos and stages of diseases. Cats and dogs to be hypoallergenic, salmon or farm fish. It's a big controversial issue, but Atlantic salmon have been genetically modified with the growth hormone gene from another species of salmon to increase their growth rate. And there are a lot of concerns about how that impacts natural populations of salmon. And then, cows have been genetically modified to produce milk with the same composition as human breast milk, which is an interesting application. We're going to go through the steps briefly of gene therapy. So the normal gene is isolated from a healthy subject. That gene is then cloned. The gene is then inserted into a viral vector or virus vector. Then a bone marrow sample is taken from a patient with whatever genetic defect is being treated. Then stem cells from the bone marrow are transfected with the virus. And the transfected cells, which are the red dots, are infused into the patient. And the patient is observed to see if they actually express the normal gene. Those are the steps. Those are in your PowerPoint that's in the course shell for you to look at. So also in section 10.4, we talk about different types of genome analysis. DNA profiling is what the technique is called. Organisms have unique DNA sequences, and so DNA can be analyzed by identifying specific genetic markers or combinations of DNA sequences that are unique to each individual. And then, so the unique DNA sequences are called polymorphisms, and that just means many forms because they vary within the population of a species. And the exception to this would be identical twins. They're going to have the same DNA sequences. So techniques used in order to analyze genomes include PCR, which we talked about earlier. That's just amplifying DNA to increase the number of copies of genes that you have. DNA gel electrophoresis to separate fragments of different lengths. And then the use of hybridization probes or primers to identify specific sequences within a genome. The most reliable methods of DNA profiling are called STRs or short tandem repeats, mitotyping or mtDNA, or single nucleotide polymorphisms, which are known as SNPs. And so short tandem repeat involves, okay, so these are common polymorphisms, short tandem repeats. And what they are repeated sequences of the same nucleotide sequence. And single nucleotide polymorphisms are, I should go back to the second slide. Single nucleotide polymorphisms, or SNPs, are variations of a single nucleotide at a given locus or location on a chromosome. And then mitotyping uses mitochondrial DNA. Mitochondrial DNA also has sequences unique to a given eukaryotic species, and that can be useful for tracing family history and ancient origins, and also in identifying species of animals, plants, and fungi. And finally, without going into too much detail, there's a technique called microarray analysis, which, in contrast to other techniques which only allow scientists to track 2 or 3 genes at a time, it allows scientists to track the expression of many, many, many genes at once. And so it also gives insight to scientists about which genes are actively transcribed in a cell under different conditions, like health versus disease or growth versus differentiation. And so that has been a very powerful tool for scientists to be able to track the expression of thousands of genes in a single cell in a single experiment, instead of having to do a lot of different experiments to track this many genes. You would have to do a lot with the other techniques. That's it for chapter 10. Thank you for listening.